In a laboratory, pharmaceutical development happens in milligrams. A chemist works with a few grams of active ingredients to perfect a single dose. This lab-scale work fits the academic definition of pharmacy, the art and science of recognizing, preparing, and dispensing compounds for the mitigation and cure of diseases. But multiplying a 10-gram recipe into a 1,000-kilogram batch creates physical failures. Gravity and friction act differently on a ton of powder than they do on a handful. A formulation that is perfect in a beaker can become an unworkable sludge when scaled up linearly. This shift in volume stresses three specific safety pillars. Physical safety requires the tablet to maintain its shape. Chemical stability ensures the active drug doesn't degrade into impurities. And biological safety requires strict limits on the microbial load, or bioburden, allowed in the batch. For a drug to be considered safe, it must cause no unacceptable harm at normal therapeutic doses while remaining therapeutically effective. To maintain this standard across millions of doses, industrial pharmacists abandon the giant beaker approach. Instead, they use engineering steps to isolate and control every physical variable of the material. The industry solves these scaling challenges through 5 primary drivers: economic, accuracy, greater scope, changed character of medications, and nationally standardized formulae. The first driver is economic. As manufacturing batches increase in size, the per-unit cost of production decreases. This mechanical efficiency generates the revenue needed to support dedicated analytical laboratories that ensure the accuracy of every dose. Scale also provides greater scope. As noted here, therapies like antibiotic production are physically impossible to prepare on a small scale. They require industrial-scale bioreactors and 1,000-liter extraction columns to be viable. This centralization eventually replaced traditional extemporaneous compounding, the hand-mixing of drugs by local pharmacists. Proprietary products manufactured in large facilities created standardized national formulations that ensure a patient receives the same medicine regardless of where the prescription is filled. Scaling up is a transition in how medicine is standardized, moving from individual craftsmanship to global industrial consistency. To control the variables of mass production, engineers organize the factory floor using the unit operation method. A unit operation is a distinct physical step causing a change in state or composition. We can see the core 5 listed here: mixing, size reduction, filtration, drying, and compaction. These steps are mapped on a flow sheet, defined here as the breakdown of a sequence of unit operations. In the mixing operation, for example, the equipment must match the material's density and viscosity. If the speed or blade type is incorrect for the material's weight, the ingredients will separate rather than blend. Viscous, wet granulations require a ribbon mixer with helical blades, while dry, free-flowing powders are blended in V-shaped mixers that rely on geometric tumbling to achieve a uniform mix. By segmenting the process into these specialized steps, engineers isolate variables like shear force and blend time. This allows them to achieve the same physical uniformity in a ton of material that a chemist achieves in a beaker. Fine powder is difficult to handle at scale. It is dusty, prone to separation, and does not flow predictably into high-speed machinery. The solution is granulation, the process of agglomerating fine dust into uniform, heavier granules that flow like sand. In wet granulation, liquid binders are injected, causing irregular dust to clump into uniform spheres, the standard procedure for most tablet formulations. If the drug is moisture-sensitive, engineers use dry granulation, squeezing the powder into dense sheets before milling it back into granules. Wet granules must then pass through a drying operation, typically in a tray dryer or fluid bed dryer, to remove solvents and stabilize the granules before they reach the final compression stage. Granulation alters the physical geometry of the drug so it can survive the high-speed mechanics of industrial transport and compaction. This is a rotary tablet press, the critical point where loose granules are subjected to thousands of pounds of pressure and shaped into solid doses. The granules flow from a bulk storage hopper, through a feeder, and into the die cavity. Inside the cavity, the lower punch moves vertically to dictate the powder volume. Because powder density is uniform, this lower punch position strictly determines the tablet's final weight. The upper punch then drives down. Its depth determines compression force, dictating tablet thickness and crushing strength. Most active drugs cannot survive this pressure alone. Formulations require excipients, inactive ingredients like binders and diluents, to absorb the mechanical force and lock the tablet together. They also require lubricants like magnesium stearate, which prevents the compressed powder from welding to the steel punches during the high-speed rotation. The tablet press translates mechanical position into a precise pharmaceutical dosage. To prove the biological efficacy of the drug hasn't been compromised, the industry uses a dual oversight system: quality assurance, which focuses on process prevention, and quality control, which focuses on product detection. Quality control utilizes durability tests. To test chipping resistance, tablets are tumbled in a Roche friabiliator, needing less than 1% weight loss to pass. The disintegration test measures physical breakdown. Tablets placed in 37-degree buffers must break apart within 15 to 30 minutes. Finally, the dissolution test measures the actual chemical release rate. Samples are taken every 10 minutes to verify exactly how fast the drug enters the solution. These tests are the ultimate proof that the stresses of mass production haven't damaged the drug's delivery mechanism. All of these operations are divided across 5 industrial departments: R&D, production, quality control, regulatory affairs, and marketing. They operate under Good Manufacturing Practice, or GMP. As seen in these requirements, GMP mandates facility sterilization and department separation, ensuring that different medications, like antibiotics, are never cross-contaminated. These standards are harmonized across the US, Europe, and Asia by the ICH guidelines. These guidelines, used by agencies like the FDA and EMA, include specific focuses on quality by design and quality risk management. Enforcement occurs through on-site inspections. Any deviation from the approved procedure triggers a mandatory corrective and preventive action, or CAPA, implementation. Without perfect compliance and documentation, a drug is legally deemed adulterated and cannot be sold. The industry is currently in a transition toward continuous manufacturing, moving away from traditional batch processing to an integrated non-stop flow. This shift is driven by Process Analytical Technology, or PAT. Advanced sensors monitor fundamental quality attributes in real time as the drug moves through the machines. PAT blurs the line between QC and QA. By catching deviations as they happen, the system can self-correct, which drastically reduces material waste. Industrial pharmacy is the practice of controlled scaling. It is the application of engineering and law to ensure that medicine manufactured by the ton remains as safe and effective as the milligram perfected in the lab.